RNA or ribonucleic acid. So there are different kinds of RNA. So the first one I'm going to go over is messenger RNA or mRNA. So mRNA, it has the information to make the amino acid, to make the protein. So the mRNA codes for the amino acid sequence that the ribosomes need to create that protein. So it's made like DNA replication is made 5' to 3' from the DNA template, and it's done by RNA polymerase in the nucleus. And like I said before, instead of thymine that's used in DNA, mRNA uses uracil instead, so U instead of T. And just like DNA replication, the new strand made is complementary and anti-parallel to the template. But before mRNA can code for the protein synthesis, it needs to undergo post-translational modification before being released to the nucleus and then being used by the ribosome in the cytoplasm for protein synthesis. So the messenger RNA, it has 64 codons. It's a universal and ambiguous code. So eukaryotes and prokaryotes, they both use this mRNA codon. But each codon, even though there's 64, it only codes for one specific protein. Amino acid. So each of the 64 codons, they code for one and only one amino acid. So mRNA is monocistronic in eukaryotes. So that means each mRNA molecule so each mRNA molecule translates into one protein. Sorry, that's so each mRNA transcribed is only translated into one protein. But it's polycystronic in prokaryotes. So that means depending on where you start the RNA transcription, it results in a different protein. So since prokaryotes, they usually have a lot less DNA, only one ring of DNA, they only have a small amount of DNA. So they evolved to be polycystronic. So depending on where you start transcribing RNA from the DNA, it results in a different protein. So like you see in this picture on the bottom of the prokaryotic DNA, when you, prokaryotic mRNA, when you transcribe from one position compared to a different position, these two positions, they create two different proteins, while eukaryotes, each mRNA sequence, it will only create one specific protein. And then the next kind of mRNA, next kind of RNA is transfer RNA, otherwise known as tRNA. So this converts nucleic acid language into what I call amino acid language. So this is the part where you're actually making the protein. So you're using the information from the DNA and RNA and converting it into amino acids. So the structure of tRNA, each tRNA has a folded RNA strand with a 3-nucleotide anticodon. So this means that this anticodon, it's opposite to the mRNA. So the anticodon pairs with the codon on the mRNA. And that's how the ribosome uses tRNA to create the protein, by converting the codons on the mRNA into amino acids that are bonded to the tRNA, and it recognizes which amino acid it needs to use because the tRNA has the anticodon bounded to it. So you can say that the tRNA is charged or activated when it's connected to an amino acid. So sometimes in the cytoplasm, there will be unactivated or uncharged tRNA, so it doesn't have an amino acid bounded to it. But once an amino acid that correlates to the anticodon is bounded to it, it's called charged or activated, and it can be used in protein synthesis. Sorry. Yeah, and like I said before, transfer RNA is in the cytoplasm, where protein synthesis is happening.